before I forget though, if you have not sent me your clicker numbers yet, please do that, even though there not going to be any today. I've posted most of the people who have sent me their clicker numbers already, but please go ahead and do so. I've got all the scores, just eventually they will get uploaded. What's happening on Monday? We have a midterm, exactly. So there is a class in here right after our class. If you need more time, we're just going to head out in the lobby and keep working. So there should be no time pressure at all. Will be, when I get it done, 50 multiple choice questions, the big blue Scantron. So questions about the exam? Those of you who had me for molecular, it's pretty similar format. Hopefully, we'll have a little bit of time at the end. Again, it depends on how many questions you ask. I brought all of my toys, or some of my toys, I should say, here, particularly to look at T numbers and icosahedral symmetry. So that seems to be a concept that is always challenging, and this year seems to be no difference whatsoever. So, and as usual, again, for the people who haven't had my classes before, these are things that I wanted to re-emphasize from some of the lectures. There's no guarantee whatsoever that they will be on an exam. It's also a way for me to go back and try and talk about something again that I think I did a really crummy job of explaining the first time around, maybe because it's the end of the lecture or whatever else going on, or I actually made a mistake, which would not be the first time and won't be the last. So I wanted to start out with this whole concept of viruses versus virions. And I was just listening to This Week in Virology this morning, and they had a nice discussion about whether virions were viruses and mitoviruses, things that don't have capsids are viruses. And this is where we actually have a difference of opinion. So at least as far as this course is concerned, we're defining viruses as those that make virions and have an extracellular form. There are definitely things that replicate that look a lot like viruses but don't have any kind of extracellular form. And this is kind of like we've talked about multiple times already. It's biology. Biology doesn't have names for things. It just has things. And we come up with names for them. So there are always going to be some gray zones a little bit in between. So the first 5 minutes of the podcast is actually really quite nice, talking about the sort of gray zones and things. But yeah, here, the take-home message from this is that I do this all the time. Many other people do as well. Don't mistake this virion for a virus, because there's plenty of times in a virus life cycle where there's no virion whatsoever. Here on the outside of the cell, here when the genome is being replicated on the inside, and then eventually starting to make virions again. This particular cycle has lysis, but again, the message here is really that virion is not virus, even though I will make that mistake many, many times throughout the rest of the course. But just don't conflate these two together. And again, by the way, stop me if there's anything here that you'd like me to spend some more time talking about. Viruses infect everything, pretty much any cellular organism, and again, depending on your definition of viruses, even some intracellular compartments. In fact, mitoviruses was one of the things that they were talking about on TWIV. So viruses which infect mitochondria. So pretty much any cell, any organism has viruses associated with it, be they present in their genome or some kind of extracellular particle, again, virion, which is known to infect these particular organisms. And so really absolutely everywhere. And viruses are really important for all kinds of fundamental issues about what we know about life in general, things like evolution, gene transfer, etc. So that, and of course, huge amounts of our genome, again, depends on how you count the various different pieces. And this always brings me back to this slide. I keep talking about the little dots. Seem to always keep coming back to these. Way more virions in any environment, our blood, our guts, the oceans, the air, than anything else. So massive numbers of virions that are really present in all environments. So it's not surprising that they have a very large influence on what's going on in terms of life in all of those different environments. now that we're all completely clued in on T numbers and Nacho's lecture was completely clear, these images will hopefully make a lot more sense in terms of the virions. In terms of their icosahedral symmetry, one of the things that I have a tendency to really lean on even more as far as icosahedral symmetry is concerned is their similarity to a sphere. And it's just basic geometry that you have the smallest surface area with the largest volume. The fact that we use nucleic acids as our genetic material means they're actually pretty inefficient in terms of space. And so they've got to get packaged into a particle. Again, a virion, as we're defining viruses as having virions here. So something that's close to a sphere is going to be really good. And how do you make a sphere with repetitive subunits? You make something that has icosahedral symmetry. And so that's a really nice way of getting these, again, large volumes but small surface areas. The difficulty, of course, with this is that all of these virions have to also be able to release the virus genome, and so the metastability state. And we will talk more about these plant viruses a little bit later on with these probably bacterial viruses that were just found in an environment. These tails are almost certainly involved in getting through membranes, and we talked about a couple of those already. And then we also have the helical forms of your virions. And then again, it's biology, so we have exceptions like this guy here, and of course, even more exceptions in archaeoviruses that we'll talk about right at the end of the term. So this is the plaque assay, also very important concept. Really hard to write exam questions on plaque assays, but I'm doing my best to come up with some good ones. But the basic idea here is it's a way to count infectious virus particles. And that's really the key to the plaque assay, is you're looking at infectious particles. And many of the other techniques, even the fancy diagnostic techniques that you'll see in doctors' offices, hospitals, etc., these are usually based on either counting virions, which may or may not be active and infectious, or counting genomes, which may or may not be infectious. Those are way easier to do than these plaque assays. But if you really care about the number of infectious particles, you have to do some kind of infectivity assay. And yeah, sure, you can use animals, but those are a lot harder to work with and way more expensive. If you have any kind of cell culture system, you really want to do these kinds of plaque assays. So a couple of key aspects about these plaque assays. The first one is a dilution series. So you start out with your concentrated virion, or in that case, it could even be infected cells here. Do dilution series, and the actual numbers of this dilution series really don't matter, and then take some amount out at the end and put it onto a set of non-infected but susceptible and permissive, and we'll talk about susceptible and permissive later on, means that they can actually now replicate these viruses. The fact that you've done this massive dilution means that you have way fewer infective viruses than you have host cells. And that's really important for this plaque assay. And then, once you've done this, you incubate, and this incubation step, again, is also critical because what it does is it allows the virus to replicate, to spread to the host cells that are next to it, and then those host cells to produce more virus to spread to the cells that are next to that. And the reason that this has to be done, you have to have something which is detectable. You did a detectable infection of some way, shape, or form. And usually that's killing off the cells or producing some kind of marker that can be detected in an external way, maybe an antibody to one of the virion proteins, for instance. But you're not going to see one individual cell with one virion that has infected it. You're going to look at a whole set after, usually it's anywhere between 5 to 10 rounds of replication that you'll see in terms of these plaques. So then you have to detect them. In the case of a lytic virus, particularly lytic viruses of bacteria, You have bacteria that grow really nicely in the background here, and then you just have the holes where they have been eaten, because it's a phage, which is eating the bacteria. And then each of those holes corresponds to where you had one virus infection back here before your incubation. You know what your dilutions were, so you can back calculate how many plaque-forming units or infectious virus particles that you have back here in your virus suspension. So is that clear for plaque assays? Again, this is an important concept, and the TWIV guys and gals are all gung-ho about plaque assays because it's a really way to look at infectious particles. It doesn't work with all viruses, unfortunately. Would be nice if it did. So one of the things, just one of the things that these plaque assays are useful for is doing one-step growth curves. And the whole idea of a one-step growth curve is sort of like taking this plaque assay and kind of flipping it on its head. Instead of having really few numbers of infectious viruses and a vast excess of host cells, now you want to have a vast excess of viruses and a smaller number of host cells. And the reason for that, you want to do a synchronous infection. And it gets back to this same issue with the plaque assays. You can't see one virus infecting one cell. You want to look at a population in order to be able to have enough that you can detect. And detection limits are getting better and better, so you can do fewer and fewer of these synchronously infected cells and a virus replication cycle. So this is supposed to represent what each individual virus replication cycle looks like, but it's looking at a whole population of them. So you do these synchronous infections at high MOIs, multiplicities of infection. I wasn't planning on going over the Poisson distribution, but if people want to, I'm happy to. If I give you a Poisson distribution question on an exam, I will give you the equation, and you should not need calculators. So I'll give you all the numbers that you need. So high multiplicity of infection, which, again, given this Poisson distribution statistically, should give you close to 100% infection. And so that's the whole idea here of simultaneous infection versus a really, truly synchronized infection. And a synchronized infection is what you're looking for. But you try and get it pretty close. And then all you do is just wait. And then at certain times, you'll take a sample from this mixture that you have growing and do a plaque assay on that. And that will say, at this particular time, you had 10 PFUs per mil. And this is this black line now. And way out here, 30 hours later, you have 300 plaque-forming units per cell, for instance. And this is just an example of one particular virus. We'll talk about the polyomaviruses after the midterm. And you can also look, all we first look at is this black curve. That's your standard one-step growth curve. But in this standard one-step growth curve, you have this period before virus particles are being produced. And that is we're going to spend, again, most of the time talking about for the rest of the time. We already talked about this quite a bit already. You're going to have your early genes being made and your late genes being made. And these you can detect now instead of doing a plaque assay, now by looking specifically for proteins, or looking specifically for some kind of activity, or looking specifically for some kind of nucleic acid. And in the particular case here, the dark orange line, that's now your viral genome. In this case, it's a DNA genome. And all of these things are starting to be made before you actually start to make these infectious virus particles. So the one-step growth curve, first starting out, again, looking at infectious virus, that'll give you an idea what the time frame is that you need to do to look at all the interesting molecular stuff which is going to happen before you actually start to see virus particles. Happy? Yes? No? Good? Good on one-step growth curves. I did put the original publication for one-step growth curves in D2L. It's a little heavy reading, to be perfectly honest. They're physicists, and we're not necessarily writing for biologists. But if you're interested, I think it's really fascinating and the amazing insight that they had in terms of thinking about, again, the statistics and everything that went with that. So, again, all the interesting stuff is going on between your infection and the virus particles being produced, at least as far as the molecular aspect of virology is concerned. And we've talked in general about binding to receptors. So the receptor is just some cell surface molecule that the virion is going to interact with. Once you have this interaction, somehow the genome needs to be released. And this is the entry and uncoding step. And again, we talked about this in general. And we just started to talk about it for the different viruses. And we'll again continue to do that later. In general, and I don't see Echo here give me a hard time about this, early genes are those which are going to be important for replication of the viral genome. And of course, we have exceptions to all of these things, like the small RNA phage, where you get the coat protein made first, replication of your viral genome, and then you start to make the structural proteins, which are going to end up making your virion, put that virion together, and then somehow it needs to get outside of the cell. So not so much for this midterm, but certainly for some of the later ones. Each of these steps for each of these different virus families can be a little bit different. And so in terms of review, it might be good to sort of have this image and repeat it every single time for the different viruses in terms of thinking about them and that whole process. So these, again, this is all still overview. Baltimore classification, again, I don't expect you to remember that, class 1 are double-stranded DNAs, that there are 6 different classes, and in fact, some people have added a 7th up here, which is also reverse transcriptase-based replication of viral genomes. So at the very ends of each of these are the genomes which are actually being packaged in the virions. All of them have to get to messenger RNA in some way, because no viruses to date have been found that have ribosomes. And again, as soon as we discover some, because it's biology, and there probably are some or something, the gray zone again comes back. But as far as we're concerned, all viruses require cellular ribosomes. If they require cellular ribosomes, they've got to make messenger RNA somehow. And how that happens is going to be different for these different Baltimore classes. Double-stranded DNA is easy because you have either a cellular or a viral DNA-dependent RNA polymerase. If you've got a positive strand RNA, this can be translated directly. Or in order to make genome, it has to go through a negative strand RNA step. Negative strand RNA, you just make a copy of this RNA and you end up with positive strand RNA. Double-stranded RNA, separate one of the strands, you end up with RNA. The difficult ones are these retroviruses and the single-stranded DNA. Single-stranded DNA has to become double-stranded before messenger RNA is made from it, because all DNA-dependent RNA polymerases that we know of only work on double-stranded DNA. None of them work on single-stranded DNA. Really fascinating thing. If somebody could discover one, it'd be really cool. But so far, none have been discovered. So you have to go from single-stranded DNA to double-stranded DNA, and then to your messenger RNA. Retroviruses have some kind of reverse transcriptase step where there's an RNA intermediate. Again, in the classic case of the retroviruses, they package positive strand RNA. That becomes a negative strand DNA, which becomes double-stranded DNA, and then becomes messenger RNA. And in some of the other cases, here, the class 7s, you'll have DNA that becomes RNA and then goes to DNA before you make your messenger RNA. But the classic case, and if I were to ask you a question about the Baltimore classes over here, it would be, how many Baltimore classes are there? How many classes did David Baltimore come up with? The answer would be 6, not 7. Even though in some textbooks people will say there are 7 Baltimore classes, but I would say that there are 6. Which ones are which, again, I don't expect you to remember. So there are questions on the Baltimore classification kind of thing. It's just a way of thinking about different viruses, again, particularly in terms of how they replicate. And that's the focus of what we're talking about. Okay, so now we have our genomes. What do we put them in? We put them in capsids. And so this gets us to quasi-equivalence and triangulation numbers, etc. A number of people have asked me about this, so I wanted to spend a little time talking about it. So the classic, again, icosahedral symmetry. This is, trying to get ourselves close to being a sphere. You have a 20-sided particle, each of which are triangles. And the simplest that we found in terms of ways of making triangles is putting 3 subunits together. And so that's the simplest. That is actually the equivalent icosahedral symmetry, which is shown up here. So you've got 3 of these individual subunits per face, and then 3 times 20 is, of course, 60. So one thing the T-number tells you is how many capsid proteins you have in your icosahedra. Now, the whole concept, however, of quasi-equivalence, which is Casper and Klug, and to some extent Buckminster-Fuller, was that you can extend this just by adding similar, which is the quasi-equivalent formations of these capsid proteins. And so my favorite example is the soccer ball, because you have pentagons, which are your 5-fold axes of symmetry, and hexagons between them, and so it's 5 capsid protein subunits and 6 capsid protein subunits. And what Casper and Klug came up with was using this whole background here, which is the hexagonal packing. If you take out one of these subunits in a hexagon, you end up with a pentagon, and that gives you the curvature which you need in order to make a closed compartment. And so here, with one hexagon, which is going from right in the middle of one of these triangular faces, you now have 6 subunits in addition to the ones, which you had the 5 at each of the corners. And so this 6 plus 3, because it's the divisions of each of these in each of the corners, now we'll have 9 subunits per side. 9 subunits per side times 20 is 180, or you can take the T-number, which is 3, times 20, so 3 times 60, excuse me, and you end up with 180. So the T-number also tells you how many of the capsid proteins you have on any given side, and then, of course, since you have 20 sides, you're going to end up with the whole particle. The other thing that the T-numbers show you is the number of different kinds of interactions that each of these proteins have. And that, I wanted to sort of zoom ahead and look at this. So T equals 1, these are all completely equivalent interactions. Each of these capsid proteins interacts with all the other ones around it in exactly the same way. So one conformation that you have. Now if you go up to T equals 7, there are 7 different conformations that each of those capsid proteins has with each other. And that's supposed to be shown here, which is these sort of different shadings going from yellow all the way to red. So if you had something like this, how would you figure out what the T-number was? You need to get from one 5-fold axis of symmetry to the next 5-fold axis of symmetry, which you can do on this scale. I think it's easier, and most of the people who come and talk to me actually like the 3-dimensional models a lot better, because it's a little bit easier to see them on there. But all that you're doing is you've got to hop from a 5-fold axis of symmetry to the next 5-fold axis of symmetry via each of the hexamers that are in between. So here's a 5-fold axis of symmetry, here's a 5-fold axis of symmetry. You hop by going 1, 2 in this direction, which would be your H. Then you change directions. Anytime you change directions, it's a different axis. So this would be K of 1. So here you have a T equals 7. And we'll look at an example of this in just a second. So that's how we can calculate our T-numbers, just given one of these icosahedral symmetries. So here's a nice example of that. 5-fold axis of symmetry here. 5-fold axis of symmetry here. Hexamers in between. 1, 2 in this direction. 1 in that direction. H squared plus HK plus K squared will give you a T equals 7. So if I were to give you a particle like this, a picture like this on an exam, which you'll see on old exams, you would all be perfectly happy in telling me what the T-number is, right? Yes, good. Okay, and if not, then afterwards come and check out some of the models up here as well, and Come and ask me questions. I've got regular office hours today. I'll be answering emails as much as I can. Unfortunately, I need to leave at about 11:30 today, but I'll be back later this afternoon as well. So if people need some more office hours, let me know. Okay, so now we've got our virions, we've got our genome. Some virions have envelopes on the outside of them, basically a lipid membrane on the outside. How do they get those lipid membranes? They get those lipid membranes from the host. But there's one really important thing about these lipid membranes that I think I didn't talk about or make very clear earlier on. In these lipid membranes, there are virus-specific proteins, so virus envelope proteins. And all viruses that have envelopes have virus-specific envelope proteins. And what that means is that if you have an enveloped virion, it can't just randomly steal membrane from somewhere and then go off and try and infect another cell. It needs to make sure that, to totally over-anthropomorphize here, that you have the virus-specific proteins wherever that membrane is that is being taken up in terms of the rest of the envelope. So there's got to be some way of getting your virus genome, and in some cases you'll have a capsid as well as the virus genome ready to the place where all of these virus envelope proteins are. And so that's what I tried to talk about here, but I think I did a crummy job of talking about the first time around. So here, all of these orange proteins are supposed to represent the virus envelope proteins, again, sticking in the membrane, and then in this case, you have a capsid with a genome inside it. This then has to associate somehow. And sometimes you have a matrix protein, which is sort of some specific glue between your envelope proteins and the capsid. In some cases, there'll be just the capsid protein itself, which interacts directly with these envelope proteins. But the envelope proteins, again, it's really critical that you have an envelope protein, because if you don't have the envelope proteins associated with your virion, those are what's going to be interacting with receptors. And so if you don't have your viral envelope proteins, the virion, the genome that's inside, is never going to find the right cell. It's never going to be able to replicate. So that targeting is also very, very important. And this process, just a second, is also known as budding, because you have a vesicle which then buds off of the cell, and afterwards the membrane is complete for the cell. And so the cell doesn't have holes in it when these vesicles are released. And it turns out that this is not just a viral process. You also have cells which release vesicles. Classic example are neurotransmitter cells, although it turns out that some of them actually have virus-like proteins associated with them. Another cool story that we have a chance to talk about later. Yeah, Megan.
So if a virus is really affecting a host, at what stage does it
Okay, so the question, if I get your question properly here, so when are these virus envelope proteins made, and how do they get to the membrane? We'll spend a lot more time talking about that when we talk about some of the specific ones later on, but it's just like membrane proteins would otherwise be made. So through the Golgi process and secretion, et cetera. Okay, other questions? Yeah, David.
How important is exclusion of the host protein from the viral envelope?
Yeah, how important is exclusion of virus protein from host envelope? That's a really good question, and I think the answer is nobody really knows. Probably what seems to happen in most cases is that a lot of these, and I have a slide about this a little bit later on, but we can talk about it now. You seem to have kind of 2D assemblies of the virus envelope proteins in specific parts of the membrane, and that's where they then associate specifically. And so they also seem to be associating with each other. So they're probably excluding to some extent the cellular membrane proteins, but there are definitely cases where you can purify virions and you find cellular membrane proteins associated with them. So it doesn't seem that there's a complete exclusion that happens. Yeah, because they're membranes. These are pretty floppy, so it's not surprising you'll have some of that happen. Okay, so switch gears, talk about virus taxonomy, how to think about how they're related to each other. The details of this particular image are not important. What is important here is that we're looking at sequence similarity. So if you have nucleic acid, in this case sequence similarity, for that matter protein sequence similarity, you can get a really good idea how virus genomes are related to each other. And that's a really nice way of thinking about the phylogeny, thinking about where some of these viruses have evolved from. Great way of doing classification if you have some way of comparing the sequences, and they do have some kind of sequence similarity. If you don't have some kind of sequence similarity, that brings us to this idea, which is really a concept. Until we have time machines, we're never really going to be able to tell whether this is true. But that it is rather striking that a number of viruses which infect completely different kinds of hosts, and when I say completely, I'm meaning different domains of life, archaea, bacteria, and eukarya, have similar capsid proteins in their virions. And such similar capsid proteins that they're really kind of indistinguishable from each other. And so I brought again some of my models for these things. I can't even remember which one is the one that infects a bacteria, which one infects archaea, and which one infects eukarya, because the structures are so similar to each other. But the sequences are really completely different. So you can't align the sequences, and you do need to align sequences, by the way, to do these phylogenies, because you need to have some signal there. So if the sequences are not similar enough to each other, you can't do these kinds of analyses. So here, potentially, it's a protein homology. And so my clicker question that I guess I didn't explain well enough because a lot of people missed it, was that if you're thinking about really deep phylogeny, so things that are very, very distantly related to each other, potentially using these capsid proteins and their structures is a good way to think about how they're related to each other in terms of phylogeny. This, of course, gets back to that we're defining viruses as something that has a capsid. So if there's something that doesn't have a capsid, how is that related to any of these things? Who knows? So that's all I want to talk about in terms of phylogeny and classification. We have the Baltimore classification, and then the structural, and then sequence similarities. Okay. Entry, and this is kind of getting back to your idea. What's the flip side is going to be exit, but similar kinds of things. So how do you get inside a cell? The enveloped viruses have a pretty easy time because cells have membranes on the outside. So if you have your envelope and the cell's envelope, they can fuse to each other, and then you release your capsid or genome from the inside. So that's really easy. The much more difficult process is if you have a naked or non-enveloped virus, somehow that needs to get inside the cell as well. Fortunately, at least for the viruses, there are mechanisms that cells take up other things from the environment. Again, not terribly surprising. And then viruses have just taken advantage of this. And so all of the processes that you have for virus entry, particularly these enveloped viruses and many of the naked viruses as well, they're taking advantage at the very least of some kind of extracellular molecule that they bind to, again, the virus receptor. And in most cases, it'll be some kind of cellular process that's being used to bring that virion or its genome inside the cell. There are, of course, exceptions. There's always exceptions, and probably the best example of those are the bacteriophage. And so the injection, where you inject something or make a hole in the cellular membrane in order to release your genome, that's clearly not a cellular process. But here we have the fusion process, and in many cases, excuse me, you'll have back up, this receptor-mediated endocytosis. This seems to be true for the majority of viruses, at least the animal ones that we're talking about, where you have first a endosome which forms, and then you have fusion of the viral envelope with the cellular membrane, but only after it comes inside the cell. And what we've talked about a lot is that this is very often to be dependent on a pH change, because endosomes the pHs are lowered because that's what cells are usually doing to break down whatever they brought in, usually be some kind of food source. And then that change in pH is very often sensed by the viral envelope proteins or some other part of the virus in order to change the structure. This was one of the figures from that part, excuse me, of the entry chapter, basically just showing that you've got lots of different ones. And then when we talk about, for instance, HIV, it fuses at the membrane. This core is released. That needs to get in the nucleus. Flu comes in to endosomes. Adenovirus comes into endosomes. Hepatitis B comes into endosomes. Parvovirus comes into endosomes. Should kind of sound like a broken record now. Many of these virions need to get their genomes into the nucleus. This can be DNA genomes like hepatitis B or adenovirus, but also some RNA viruses need to get their genomes into the nucleus as well. And we'll talk about those RNA ones in much more detail when we talk about those RNA viruses a little bit later on. So this is, again, it's mostly in terms of review and thinking about the different ones here. And then, of course, the viruses we talked about specifically later are not the ones that are on this list. Probably the best understood of actually probably the whole entry process, at least for enveloped viruses, is what happens with flu and hemagglutinin. This is the protein which binds to the receptor but also is important for fusion, getting those two membranes to fuse to each other and release the virus, in the case of flu, multiple genome segments. These start out as one large protein, which then gets chopped into two slightly smaller proteins. HA0 goes to HA1 and HA2. We'll talk a lot more about proteolysis as we move on in the rest of the course, talk about some of these different viruses. But proteolysis is a really great way to go from something which is really stable to something which is non-stable. And so this whole idea of virions being metastable, they've got to be stable to the outside environment, but then once they get to the right place, they've got to be able to fall apart and release their genomes. So proteolysis happens here, and the important thing about proteolysis for hemagglutinin is it releases this fusion peptide. And all that fusion peptides are stretches of amino acids that have very hydrophobic side chains. And so once you have interaction with the right cell in the right place, which in the case of flu is happening up here when it binds to sialic acid, gets to low pH, which is in the endosome, you have a conformational change, and conformational change, conformational change, conformational change, conformational change, again, something lots of virions do. And that then allows you to get this fusion peptide inserted into now the cellular membrane, and then more conformational changes. They're going to pull the two membranes together. And then once two membranes are pulled together, then of course you can release your genome. Virus genome can go and do its thing. Partly because this is really the early process in how you have virus infection taking place. Not surprisingly, it's the target for lots of antivirals. And my favorite antivirals, the vaccines, what they do is they will then get your immune system to block any kind of binding. So they will grab those virions and sequester them, cause them to fall apart, etc. So if you have no interaction with the receptor, you're never going to get an infection. So that's why vaccines are so wonderful. And anyone following me on Twitter probably sees that I'm posting on vaccines more or less constantly. So, and don't get me started on pro-disease people, which is what I call anti-vaxxers. So this, vaccines are by far and away the best, but also there are some specific therapies that people use to try and also block interactions of virions with a receptor. Just literally making antibodies now artificially and using those, that seems to work reasonably well. Another possibility is to put in a whole bunch of receptors and then basically try and sop up the virions because they'll bind to these receptors that are not associated with cells, and then they get really confused and they don't know what to do and they're not in the right place to actually have an infection. This is by far and away the best way to protect against some kind of viral infection. On the other hand, uncoating is also a pretty decent target as well. And by uncoating, that's just the release of the genome. And so the release of the genome, in the case of flu, there needs to be those conformational changes. Conformational changes you can block by blocking the movement of that fusion protein, and we saw the animation of the HIV one. And then other drugs, and again, the names of these drugs are really not that important. I can't remember them. I can't expect you to remember them either. There are also ways to try and do much more general blocks to virus entry. The classic one of those is to change the pH of endosomes, and there are drugs that will do that. Unfortunately, we need endosomes for lots of other processes, so it's not a terribly specific kind of drug to use. And then, since we are still in flu season, wanted to mention the neuraminidase inhibitors, so enzyme inhibitors. And just enzyme inhibitors is some kind of enzymatic activity that you have to get entry. In this case, it's enzymatic activity that you need to get release of the virus particles. And that ties us into how virions get out, either blasting open the cell, and this is usually what happens with naked virions because they get through the membrane somehow. And usually, again, we talked about budding before. That's taking a bit of membrane with you, but if you're a naked virion and your receptor binding proteins are on the outside of your virion, you don't want to have membrane in the way. And so usually with these naked virions, they're going to be released by some kind of hole or, again, in many cases, particularly for the bacterial viruses, just completely blasting open the cell itself. As we talked about before, the really important aspect about this is making sure this happens at the right time after all of your infectious virions have been made. On the other hand, budding, which we talked about, enveloped viruses, and this is getting back to your point, David, about how you get the virus envelope proteins together. They have to associate in some way and then get your virion to them, and usually that excludes most of the host membrane proteins in the process. And then these virion-associated pyramids that we'll talk about in the very last part of class because they're so cool. Hey, sorry, more questions about this general stuff before we talk about the RNA viruses, the single-stranded DNA viruses, and T7. Okay, so RNA viruses, I should say RNA phage, not RNA viruses, because we'll talk a lot about RNA viruses after the midterm. So the RNA phage are very, very simple genomes. They have what they need to make more of their genome, a viral replicase. They have a coat protein, because we've decided in this class we're only going to call things viruses that have coats. And then a couple extra things, maturation protein and lysis protein. So lysis protein, not surprisingly, this helps the virion to get out of the cell that it's infected. And then the maturation protein serves as a receptor binding protein, and as we now know, also helps the virion to get out of the cell as well. So these are the very simple systems. There are other small RNA phage. Q beta is the best known of those. I went looking really hard to try and figure out what this A1 protein is actually doing because I didn't know what it did, and there's nothing in the textbook to talk about it. And I went and looked, and there's a paper in 1974 that says it's absolutely essential. But why it's essential is really not completely obvious. So this read-through protein, however, is interesting just from the point of view is that there's a alternate translation, and we'll talk more about alternate translation later on with some of the other viruses, particularly for the retroviruses, where normally you have a stop codon, but in about 5% of the cases, this will then actually not be used as a stop codon. You end up with a longer protein, and there's something about this longer protein probably assembles together with coat in order to get the active virion. Now, why you need that in some particular cases, again, I couldn't figure out, so can't expect you guys to remember it either. What we do know is that these virions, and this is really quite recently, are actually not nice, completely quasi-equivalent particles. They're asymmetric. They've got that maturation protein at one point, at one of the five-fold axes of symmetry, and they're so well conserved in terms of their structures. When you average thousands of them together, you can actually even see where the genome is inside the virion. So yeah, we got a very good handle on exactly how these things look as virions. When they get inside the cell, then you need to be making more genome and more virions. How that's done for these small RNA phage is really all about translation. Not surprisingly, because it's RNA, you don't have any transcription going on. And that translation is regulated in a couple of different ways. The major one is secondary structures and literally translational initiation. So translational initiation, pardon me, is going to happen at the coat protein in particular. That starts to make lots of this coat protein, and then after this coat protein is made and lots of ribosomes are chunking their way along the RNA, it changes some of the secondary structures, and then the replicase protein starts to be being made, and only can be made again when you don't have these secondary structures because the ribosome has come through here. Eventually, this replicase protein will block the formation of more coat protein, and this is important because the ribosomes and the RNA-dependent RNA polymerase are trying to go opposite directions on this RNA. Ribosomes are going to be translating. The RNA-dependent RNA polymerase is going to be trying to make more genome. And so there's always this fundamental conflict which happens there. And then the other fascinating part about how you get this lysis protein is it's not really dependent on a secondary structure per se, but it's dependent on the ribosome staying on the RNA and just moving back in a few percentage of the cases and starting to make some lysis protein. And so it's just the distance that this lysis start is away from the termination point, and everything that I mentioned about translation and translational termination last term in molecular biology is kind of moot, because everyone, I always say, okay, you got release factors, the whole thing falls apart. Well, not in all cases. Sometimes it stays on and can restart right next to it. Okay, so that's all I wanted to talk about in terms of these replication of these small RNA phage. I did want to spend a couple of minutes talking about the MDA and how people are now using particularly the Q-beta replicase. And so again, these replicases, they're RNA phage, so it's an RNA-dependent RNA polymerase that can make many copies of a particular RNA. And what this is used for is to amplify RNAs, but particularly amplify RNAs that have base paired with something that you're looking for. And so the key to this kind of experiment and this particular kind of assay is you have what you're looking for, and you know what the sequence is of whatever it is you're looking for. This has two regions that can base pair on it. One region is so you can trap it somehow. So if you have a sample, serum sample, blood cells that you think might have a small amount of HIV in them, you get all the RNAs out of those, and then you immobilize that RNA somehow. And that usually is going to be through base pairing with some kind of DNA fragment, etc. The problem with this immobilized RNA is it's really hard to detect. And so what you want to have is some way of amplifying that RNA. And there aren't any good PCR-like enzymes, DNA-dependent DNA polymerases that you can use for this. But if this can now base pair with one of these MDV RNAs, if this MDV RNA is now associated with, through this target RNA, something that immobilizes it, and you put in the Q-beta polymerase, you'll make a whole bunch of that MDV RNA, sorry. The MDV RNA then is just a marker saying this was here. If, however, you don't have any of that, you put in this MDV RNA, and the Q-beta polymerase, nothing's going to amplify. Does that make a little more sense? I think I did a really crummy job of talking about this first time around. So it's just a way of detecting that you've got something in your sample, and the way that you've detected it, you've got this MDV RNA, which can base pair to it. If it base pairs to it, it means it's there, and you use the Q beta to amplify it. Okay, so now our small RNA phage are done. Now let's talk about this single-stranded DNA phage, the extraterrestrial genome. Why was it called an extraterrestrial genome? The first reason was that no sequence naturally would ever form that has all of these overlapping and open reading frames. Well, yeah, maybe it does because all of these things are highly flexible and it's a way to invent genes. But at the time, no one thought that it was really a possibility. And then the other aspect about the extraterrestrial genome is that it self-amplifies. And so if you were to send a virus to another planet and then have a really small amount of signal that you sent, it amplifies because it's going to replicate in bacteria, something which is there. And then this extraterrestrial intelligence also assumes that you have a way of determining sequences and that you're interested in these sequences and you see that they're really different. So not a totally crazy idea, but we don't have any evidence of who sent it, and we now know what all of these things mean as far as the virus is concerned. So we don't think that it's actually a message from some kind of extraterrestrial intelligence. And a lot of that has to do with the fact that these sequences, the A sequence actually is pretty well conserved. And we know that the A protein is what we need to make more copies of the single-stranded genome because it cuts the genome and religates it. We'll take a look at that in just a second. These other open reading frames that overlap with it are actually not essential or can be incredibly heavily modified and still function. The B protein and the D protein in particular, these are those scaffolding proteins. And scaffolding proteins, they have to have a particular structure, but exactly what the sequence is of the amino acids in those structure is really not as important. And we'll also see later a number of other scaffolding proteins. Those scaffolding proteins have lots of flexibility in terms of their sequence. On the other hand, the structural proteins, F, G, and H down here, these aren't overlapped, and just because of that, you've got a pretty good idea that they're not going to be things which you can have lots of mutations that happen in them. So yes, genome from outer space. This is not an image from the textbook, but I think it's really nice because it basically gives us everything we need to know about both entry and assembly processes. So we now know, since the publication of our textbook, that we have this H protein, which forms this, I'm trying to remember what Ben Fain called it, rather inappropriate change in its structure, where you have interactions, again, first with the LPS, and then this 10-helix bundle, again, brand new structure that nobody knew about before, which then makes a hole through both membranes, and then you have the single-stranded DNA, which is released, probably just because it can't go back through this channel in the H protein, and then cellular replication. Once you have these all make its proteins, then these proteins assemble as pentamers, and together with the internal scaffolding protein B and the external scaffolding protein D make these procapsids, and in the procapsid, that's where you have the DNA gets packaged. After you have DNA packaging, then you actually lose some of your structure to end up with these final particles, which are the infectious ones. Other aspect of these single-stranded DNA viruses, which of course is very different. Oh, sorry, Diana, yeah.
So is this the one where the single-stranded DNA is being pulled through transcription?
Okay, so it doesn't seem to be through transcription. First, because you can't get any transcription on single-stranded DNA. It's got to become double-stranded in the first place. So there may be some pulling which is happening through replication that we don't know. what Ben Fain thinks is happening is it's just literally diffusion, and that it goes through here, can't go back, and then you will start to replicate it, and eventually it comes through. But again, that's not entirely clear. T7, on the other hand, that's the one where you have transcription, which is starting to pull everything through. So single-stranded DNA viruses, you need single strands of DNA. How do you get those single strands of DNA? Once you've made double-stranded DNA is all being made by the cellular processes, the cellular DNA polymerases. You have the A protein, which makes a cut in one of the strands. It then attaches to the 5' end, gives you a 3' end. Cellular polymerases will replicate their way around the genome. Single-stranded DNA binding protein will associate with this, and then you end up with, at the end of one round of replication, the A protein will religate this single strand, bind back to a new 5' end. Your 3' end is still ready to go, so you make many copies of this process. Then that single-stranded DNA that had the single-stranded DNA binding protein associated with it gets packaged into the virion. And this, I forget if it's your question, David, or somebody else's question. That single-stranded binding protein is removed, and the J protein gets put onto it instead. And so when you package, you get the J protein, which is on the inside of the particle, is then packaging that single-stranded DNA. Okay, these are the single-stranded DNA viruses. Then we have bacteriophage T7. This is the one that Diana was interested in. One thing that I forgot to mention here, again, that's why I want to throw it up again. From left to right here, this is how genome is coming inside the cell. So when you have the first binding and release, that's that first 850 base pairs, that has this transcriptional initiation site for the cellular polymerase. That starts to pull it in until you translate the T7 DNA-dependent RNA polymerase, which will now bound to all of these class 2 promoters, pull the rest of the genome in, eventually bind to the class 3 promoters, again, as they're coming inside the cell, and make lots and lots of these, particularly structural proteins, the major structural proteins here, and then some of the more minor structural proteins as well. So binds, the genome is released, and again, this is the left-hand part of the genome. First, you have the cellular RNA polymerase, and then the viral RNA polymerase that pulls all the rest of the genome inside the cell. Didn't think I did a good job of talking about T7 DNA replication. Back up here briefly. The origin of replication for T7 is way over here in this part of the genome. And so this is sort of the blow up here of those two. Well, we have first the T7 RNA polymerase that makes a primer that the T7 DNA polymerase can now extend. And this would be your leading strand here. Then you'll have a lagging strand, which is started by a T7 primase helicase. This then ends up going off to the left-hand end of the genome, the right-hand end of the genome. This T7 DNA polymerase is chugging its way along. There's only one origin of replication, but it's going in the same direction as the T7 RNA polymerase would be, because all of those genes, every single transcript, is going from left to right in the genome. Okay, happy with T7 replication? T7 vectors, wonderful tools, but the big star of the story here, T7 RNA polymerase. T7 DNA-dependent RNA polymerase, single subunit, easy to make, has very specific promoter sequences that are different than any other promoter sequence, and it's really fast. And makes lots and lots of RNA. So this is useful if you want to turn that RNA into protein in recombinant DNA processes, or also if you just want to make RNA for other kinds of studies, RNA evolution, RNA folding, etc. That's all I have. About another 10 minutes or so before the next class comes in. If people want to come and check out the structures up here, I actually have a couple of virions that have t equals 4 symmetry. Most of them are T3. I've also got a t equals 25. Good luck on Monday, and again, send me more questions if you've got more questions.